Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. The Allen Institute in Seattle is quite a unique research institution. Funded by Paul Allen, one of the founders of Microsoft, it has over the last 20 years made some seminal contributions to neuroscience by addressing problems that are difficult to pursue for university groups with more limited resources. One example is the production of publicly available transcriptomic maps showing how genes are expressed, that is, what proteins are produced in different types of brain cells in mice and humans. Over the last 10 years or so, the project MindScope at the Allen Institute has pursued an industrylab-like approach to study the mouse visual cortex at many levels of detail. A whole battery of different experimental techniques has been used: electrophysiology, optophysiology with calcium imaging, electron microscopy, often combined with the new genetic techniques offered when studying mice. Importantly, all this data has been made available for the entire scientific community, typically before the researchers at Allen have had the time to analyze the data properly themselves. A key element of Project MindScope has been the integration of all these data into mathematical models. Today's guest, Anton Arkhipov, has been leading one of the groups at Allen working on this. In a seminal paper from 2020, his group presented the first biophysically detailed model of mouse primary visual cortex, comprising 230,000 neurons. In the podcast, we talk about what the goal of the modeling was, how the neuron models were made, how the neuron models were connected to make the network model, and how the network model was tested against electrophysiological data recorded from mice watching visual stimuli. We further talk about how the model has been used since its release in 2020, how it is improved by use of new data, and how the approach possibly can be extended to model the whole mouse cortex and eventually even whole brains. Anton has many interests and recently published a sci-fi novel called Age of Cindy, which is about biotech changing our bodies, minds, and society. He has also thought about the problem of consciousness and recently presented his own hypothesis about possible fundamental physical properties underlying consciousness. We briefly touch on that in the end. And as for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoreticalneurosciencepodcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Anton. So, welcome to the podcast, Anton Arkhipov.
Thank you, Gaute. It's great to be here.
Yeah, so let's start with, you work at the Allen Institute for Brain Sciences in Seattle. I've been there, I've visited a few times. Always nice to come there. But it's quite a unique place. So why don't you tell you a little bit about it?
Sure. Yeah, it is indeed a unique place, and I feel happy and lucky to be there. It's a non-profit private research institution, which was started in 2003. So, in fact, we just had a party celebrating 20 years of the Allen Institute just a few weeks ago. So it was founded by Paul Allen, who was he was an entrepreneur, perhaps most famous for co-founding Microsoft together with Bill Gates in the 70s.
Mhm.
Um, and Paul Allen was a unique person. I had the privilege of meeting him a few times, but I cannot say that I knew him very well. But from what I understand and from what I read, he had very wide-ranging interests, and he was in particular very interested in science, biology, and especially in neuroscience, like many of us, right? He was curious about how the brain works, how the mind comes about, and what happens when things go wrong. And so apparently he wanted to spend a big part of his fortune that he acquired through his business interests on supporting research in this area. And so that's how the Allen Institute started. Now, when it first started, it was a very focused uh, enterprise, right? So there was apparently some meetings, so-called charrettes, where people who are very well established in the field would come together, and Paul Allen would ask them, so what are the most important problems that we can address, that maybe other research approaches like research labs in universities having a hard time to address. And through discussions, some directions emerged. And so initially, the directions that emerged were let's characterize gene expression in the brain, right? So there are thousands of genes that express differently in different cells. And because we have different combinations of cell types in different parts of the brain, the gene expression patterns differ between different parts of the brain. And so the initial task for this new institution was to map the expression of genes across the different areas of the brain. So that's how it started. But then after
So
several
this
years,
this was mainly mouse or humans or
yes, it started from the mouse. So the very first area of work and the first data sets that were released were the Allen mouse brain gene expression atlas. But then very soon afterwards, it was followed up also by some data sets from human. And so that established some resources that turned out to be quite useful for the community. And then after the initial several years, there was an expansion of the Allen Institute with the idea that one can try to do more and start looking more into mechanisms. And so I would say that was the last 10 years, the last 10 years of the Allen Institute. That's also when I came. So Christoph Koch, who you interviewed on one of your previous podcasts, he came in 2012, I believe. He became the chief science officer of the Allen Institute, and led this enterprise for basically 10 years. Brought in many of us there, including myself, and there was a substantial expansion of efforts. So not only studying gene expression, but also the electrophysiological and morphological properties of all the different cell types in the brain, the connectivity, and their activity in vivo in the behaving animal.
so this
Again,
was particularly the mouse visual cortex, wasn't it?
yes, it's been focusing on the mouse visual cortex, and mostly, although already back 10 years ago, basically, there was some work being done with the human tissue, and it continued. And now there is a really big research program at the Allen Institute focusing on the human and also non-human primate research.
So yeah, exactly. So it was sort of like this really comprehensive, for sort of almost industry style approach for mapping out many aspects of the all aspects almost of the of the mouse visual system, visual cortex, at all kinds of different levels of detail, and electrophysiology and optophysiology and all kinds of things, right?
Yeah, exactly. And so I think here we should also explain a little bit more about what's special about the
Hmm.
Allen Institute, right? Because there are, of course, many institutions and especially university labs that are doing research in similar areas. So I think the idea about establishing the Allen Institute was to make it something, to occupy a space, a niche that was sort of empty until then in neuroscience. So, because on the one hand, in neuroscience, we have university research labs where it's usually an individual investigator, right, with some postdocs and graduate students doing research on a particular area of their interest. And at least traditionally, all the different investigators are trying to be orthogonal to each other, as different as possible.
Yeah, so that they can get, yeah, exactly. So they make a mark and be visible and get funded.
Exactly, yeah.
Mm.
And on the other hand, there are industry, pharmaceutical companies and such that do research for profit, which is also very important, but it's much more applied, right? A lot of the times it's less about understanding how the system works, but more about developing a drug or something like that. And so the idea was that the Allen Institute can sort of occupy that space in between where it's still about basic research and understanding the system, but doing it with this industrial approach and trying to characterize the whole system and all of its
Mm.
components and how they work together. Of course, this is very important for biology and the brain, especially because things are so complex and you
Yeah.
really need to invest a lot
Yeah.
to try to disentangle it. So yeah, I think that's the idea.
And also this, as I understood it, that I mean that was like this really says emphasis of Paul Allen to really to make the data available for the community immediately, not wait until sort of like the investigators at Allen were happy and sort of looked at sort of. So it's often been a little bit ironic, I think, that I mean that's one thing is that that he pushed on sort of making this data available, but also facilitated by having these very nice web services and so on. So, because I think, I mean, I remember from the this gene map or this gene translation maps has been extremely popular, and
Mhm.
so it's sort of a little bit almost like ironic that that the data that is actually most accessible to the public are funded by a private person, while the research which has been funded by, well, typically the taxpayers, through universities and other research institutions, are typically not so available for various reasons. So I think it's really a great service to the community.
I agree. Yeah, it's a very interesting situation, right? Just like exactly as you described, and I think I'd say something about sociology of science and
Mhm.
just
Yeah,
generally how uh, certain styles of doing work and research develop, and then they sort of set themselves in stone a little bit, and you need some kind of sometimes it has to be relatively like an external push, some maverick coming in and
yeah,
trying to do things differently, and then maybe
yeah,
everyone
yeah.
else catches up.
Yeah. So how big is this institute now in the Allen Institute for Brain Sciences? You have this beautiful building on the
Yes.
lakeside in Seattle.
Yes.
So, but how many people work, or like, or roughly?
Yeah, so I don't know the exact number right now, but I believe it's something around 700 people or
Oh,
so.
wow, that's
It's
quite
a
a.
pretty large institution. It actually consists of multiple units,
Okay.
right? And so the overall organization is called Allen Institute,
Hmm.
just Allen Institute, two
I
words.
see. Yeah.
And then within that institute, there are divisions,
Yeah.
scientific units. And so there are two major ones now that work in the neuroscience space. So there is the Allen Institute for Brain Science. The
Mhm.
leader of that institute right now is Honkui Zhang. And then there is the Allen Institute for Neural Dynamics, which started a couple of years ago, and the leader of that institute is Karel Svoboda. Um, and then there are a couple other institutes that are not even doing neuroscience. They're doing, so there is the Allen Institute for Cell Science, so they're doing cell biology, and the Allen Institute for Immunology.
Yeah.
So covering multiple
So it
bases.
seems like it's flourishing. Yeah, and you are something called an investigator. That's sort of a job title at Allen, which you typically don't have at universities. That because you have your own little research group, right? Or
Yes, yes. So there are sort of different types of career tracks and types of positions at the Allen Institute, but one major ones for PIs is what we call investigator. It's roughly equivalent to a professor at a university, and so we have assistant investigators and associate investigators and senior investigators.
I see, I see. Yeah.
And yeah, the idea is that this is a person leading a research group, and it is quite similar to a professor, with the biggest difference being that we do not teach. We do not have
Yeah.
teaching component at our institute.
Yeah, that is quite unique, or maybe at least in Europe. Well, I guess at least in many countries you don't really have these. Typically, if you want to do all basic research, almost are done at and maybe at universities where people teach. But this is different. You can actually, I guess, in US you have several places, like at least a few places where you can where people only do research and do not teach.
Yeah,
Yeah, only
yeah.
basic
Maybe
research.
another example that's somewhat comparable is Janilia, right? Janilia Research Campus that also does a lot of work in neuroscience, but not only. And they have different titles there. They're called group leaders, but it's a similar idea. They do not teach typically, but focus on research.
Cool. So, I mean, this podcast is mainly going to be about sort of your work on sort of building this. Was I biophysically detailed models of mouse versus cortex? But I mean, you sort of thought maybe you could start a little bit about your trajectory in terms of how you developed your modeling interests and skills. So you started off being you're a physicist by training, right?
Yes.
And you started off doing quantum mechanics back in the days.
Yeah, that's right. So I'm originally from Russia and studied there for my bachelor degree and also master's degree at the Moscow Institute of Physics and Technology. So yeah, I studied physics. So very, there were multiple opportunities to do research, and so I started relatively early. I think after I finished my second year at the university. But traditionally, most of the opportunities that were available, at least back then, were in the area of more like standard typical physics, right? So it was less about biology, although there's definitely some strong tradition of research in biology and biophysics in Russia. Uh, but at least what I've seen most of the time was sort of more typical physics, like solid state physics, or some kind of electrodynamics and quantum theory and
That
such.
sounds like the physics I grew up with in Norway. It's like very standard, solid,
Yes,
traditional. Yeah.
yes, the old good stuff.
Yeah, exactly. Yeah.
uh, which I must say, is still it's very exciting. There is, of course, a lot of very interesting mysteries there, especially if you think about quantum mechanics and collapse of wave function and so on. Um, so, in any case, yeah, I found an advisor, a professor who was doing research in that area, and I spent a few years doing work on trying to apply sort of special non-standard formulations of quantum mechanics to simulations of some multiple body systems, right? So instead of solving Schrodinger equation, we would try to use Wigner function or quantum tomography representation to do our quantum simulations.
Yeah, exactly. But I guess one thing about this, when you do quantum mechanics, I mean, like solving this, say, many electron, like even atoms, right? The Schrödinger equation is very difficult because of this antisymmetric requirement and stuff. But
Yes.
I guess one thing which is easier than when you now are in neuroscience is that you know the parameters, like you know the electron mass and the Planck's constant. There's not too many unknown parameters there. It's just very hard. You know all the parameters, but it's like the problems lies elsewhere.
Yeah, exactly. It's sort of the opposite of the situation that we have in computational neuroscience to some extent, right? Because the problem is, you know, the equation, you know, you know what you put in the equation. Now you need to solve it, and you can solve it for 1 degree of freedom, maybe 2 or 3 degrees of freedom. But then if you have 10 particles in even 2 dimensions,
Yeah,
it's just
exactly. Yeah.
explodes exponentially into your face. And so, like, what
Mm,
do you do?
mm,
Whereas in neuroscience, we have in principle, we have the compute power to solve equations for thousands and potentially
yeah.
millions and even billions of neurons. But we just we need that structure. We need the interactions between them to simulate. So it's much more of a data question.
Yeah. And then you moved on. I think I went to US, took a PhD in US, and then you went sort of like one step closer to complexity in some sense, right? Because you started with something called molecular dynamics simulations. So what is molecular dynamics simulations?
So I came to the US in 2004 and started my PhD at University of Illinois in Urbana-Champaign. I worked with the professor named Klaus Schulten there, who unfortunately passed away several years ago. And yes, we were working. So my PhD is still in physics. I was at the physics department, but what we were doing was to apply physics methods to
Yeah.
solve biological problems. And in this particular case, it was using molecular dynamic simulations. In a nutshell, it's very simple. So molecular dynamic simulations is you take atoms that form different molecules, and you solve their equations of motion, which in most cases, or in most typical case, you simply represent as the second Newton's law.
So it's
Right.
classical. Assume classical physics. Yeah.
It's
Yeah.
I mean, there are approaches for sure that try to apply some kind of semi-classical, partly quantum representations. There are approaches where maybe part of your enzyme, right, is simulated using quantum mechanics to capture accurately the chemical reaction. But what we were doing, and what a lot of people in that field are doing, are using just standard classical equations of motion. Um, and the typical project in that field would look like this. uh, you would want to study a protein, right? Some protein that does some kind of function in the cell. And back then, at least, if there was no crystal structure of that protein available, then you are basically out of luck. Nowadays, so especially recently with AlphaFold and things like that, you may just take a sequence and create that structure, and have a
We
pretty.
just tell a little bit about like protein consists
Mhm.
of a chain of amino acids,
Yes.
and amino acids are just a well collection of some like like some particular collection of atoms, like with carbon and like or
Yeah.
like the usual suspects or oxygen
Yeah.
and hydrogen or whatever. These
Exactly.
like these organic molecules or yeah.
Yeah, exactly. Yeah, and
Mm,
then they fold in a particular three-dimensional structures, which carry out a lot of the jobs in the cells, right? So, whenever something goes wrong in disease, or whenever you are interested in a particular function in cells, or how cells interact, or how drugs act on cells, In many cases, what happens is that there is a particular protein involved, and this, what it does with its three-dimensional structure and how it does it dynamically may be important for those problems.
Yeah.
And so that's what one tries to understand with molecular dynamic simulations. You take a three-dimensional structure of your folded protein, you hit run on your computer, and then it simulates step after step. The typical time step in this simulation is 1 femtosecond.
10 to the minus 15.
15
Yeah.
seconds, yes.
Yeah.
Or maybe a couple femtoseconds. And then, at each step, the atoms move a little bit. You calculate the forces that they exert on each other. And those forces are usually things like van der Waals force and electrostatic interactions, right?
Because that's not so easy to know how these forces depend on position. That's not so easy either, right? Because you have all these water molecules around, and right. So so these force expressions, are they typically sort of come from first principles, or are they fitted to some kind of?
They are approximations. They are approximations. There are a lot of heuristics. The parameters are also for those potentials that have been used. The parameters are fit to certain observations. And the point about the water is very important one, because usually what we simulate in that field is about 80% of it is water. Right. So, you take your protein, of course, in a crystal structure, it's usually just the atoms that belong to that protein. But in the cell, that protein would be in the aqueous environment, would be in water with some salt in it, right? And so you have to put that, you create a box of water. There is the protein in the middle. And then you simulate, and 80% of what you simulate are actually water atoms of water.
So but also, I mean, I guess you could you could use to simulate I mean, for those of us who sort of know what the Hodgkin-Huxley, like how they model the ion, the currents going through these sodium and potassium ion channels. That's also proteins curling back and forth between the membrane. So I guess these things you can simulate with molecular dynamics, right?
Absolutely, yes, and that's exactly what people have been doing. At the time that I came to that lab, actually, the crystal structure of some of the ion channels were just recently discovered, and so people in that field, in molecular dynamics field, were very excited about it. They were starting to simulate these membrane proteins and the ion conduction through those proteins. And it's really remarkable that you can see that in action. You can learn a lot from the static crystal structure. You sort of know where all the different amino acids are, and you can deduce how they carry out their function. So in the case of, say, voltage gated ion channel, how they conduct an ion of sodium or potassium across the membrane. You can see the pore through which this ion is supposed to go. In the crystal structure, at least in some of them, you actually see the ions in that pore, right? Sort of caught in the act. But then what you can do with the simulation, you can take that static photo and turn it into a movie. And then you start seeing how things move, right? How this ion, let's say, conduct through the ion channel. And most interestingly, of course, you can do manipulations. You can, for example, for ion channels, in principle at least, you can apply different voltages across the membrane and see what it does to the channel. You can use different mutations on the protein and run your simulation and try to understand what happens. So it's a very, very exciting and powerful tool.
But I mean, I guess if you want to simulate the opening and closing of a channel where the time scale is maybe milliseconds or longer, then if you want to have a time step of 10 to the minus 15 seconds, and then you want to run, say, a second, that's 10 to the 15 steps. That sounds like a lot of work for even the best supercomputers.
Exactly. And that was, and to some extent probably still is, the main problem for those simulations. Because, yes, so again, this is somewhat different from what we have in neuroscience. In that field, we know the structures, at least for the proteins that we want to study, for which crystal structures are available. we know the structures. We know pretty well where all the atoms are and how they are positioned with respect to each other, right? We also know the interactions between them. It may not be perfect, but we have good approximations for it. And we can run simulations. But we cannot run the simulations long enough because exactly like you're saying, you can run the simulations, let's say, with hundreds of thousands of atoms with a time step of 10 to the minus 15 second. And you can parallelize them on a computer, right? So there are powerful software packages that
Okay.
allow to do that, but you cannot parallelize it to infinity. So at least back when I was doing it, that the rule of the thumb was that once you hit about 1,000 to maybe 100 atoms per CPU, per CPU core, you do not gain any further speed up beyond that.
So wait a minute. So you cannot have. So you cannot have. Okay. So you cannot. So you cannot speed up by splitting up 100 atoms on several cores. Is that what you're saying?
Yes, because
Yeah.
once you
So
get that few atoms per core, the computation is dominated by the interactions between the cores, right? There
yeah,
are things
exactly.
that
So
atoms do that
yeah,
are on the same core, and then there are things
yeah,
that they do between each other. And so the
mm,
more you partition, the more
yeah,
interaction you have to capture, and so then that slows down your parallelization.
yeah, exactly.
And so once you reach that sort of limit, at least back then, the typical performance was that you would get something like 1 nanosecond per day,
Wow.
right? So maybe
Exactly.
you get your million time step simulated. So now you get 10 to the minus 9 of a
Mm,
second,
mm,
right, in 1 day of simulation.
yeah.
So then, if you're a graduate student and you want to publish your paper, you can
Yeah.
run your simulation maybe for 10 days and
Mm,
maybe for a month,
yeah.
maybe for a year. I think the longest simulation I ran took about 9 months or something like
Oh,
that.
really?
Yes.
so
It,
so
it
then
wasn't.
I hope you had put in the right parameters so you didn't sort of no after
Yes.
9 months. Yeah.
Yes, that happened too, maybe for shorter simulations, but
yeah.
yes, sometimes you
yeah.
find out after a couple months and like, all
Yeah.
right, I have to start over.
yeah.
Yeah, and you run those simulations and then you get maybe 10 nanoseconds, maybe 100 nanoseconds, and you can
Mm,
learn a lot from that already. You can start seeing some movements, but a lot of what proteins do is happening on time scale of more like a millisecond, right? So we were still like 3 to 5 orders of magnitude slower than we want to
mm.
be.
So you said so when did you do this? This like 10 years ago or 15 years ago or something or
Yeah, about 15 years ago,
yeah,
like
it's like 2010.
2004 to
Okay.
2013. Yeah.
Okay, so it's a little bit better now, but maybe not many orders of magnitude better probably.
Right, right. Yeah, from what I hear, there are some improvements, but still,
Mm.
for standard molecular dynamics on standard supercomputer, you would be limited to some
Mm.
hundreds of nanoseconds, maybe tens of microseconds
Okay.
simulations.
But of course, with this experience in your bag, then when you came to Alan, and then you started this and decided to make this model for the like a primary visual cortex in mouse, which is sort of yeah, so it's quite an ambition or quite a project. So why did you decide to make this model?
Uh, well, that's actually, I must say, it's not a question to me personally. It's more a question to the Allen Institute as a whole,
Mm,
or even to our leadership, right? Because the idea with this new expansion of the Allen Institute that was happening a little bit more than 10 years ago, and that I was a part of, as we discussed a little earlier, was to try to characterize the whole system as well as we can,
mm,
and to produce some kind of understanding of what's happening there and what mechanisms are at play.
mm,
And so the idea was that, a brain, like any other system, it has, one can think about it as from sort of three major points of view. One being that, it's a physical system that has some components, right? So what are those components? What are their properties of all these components? So in the case of the brain, these are different neurons and also non-neuronal cells. So what are the different cell types that are there, and what are their properties? Let's say morphological and electrophysiological properties. Then secondly, it's a system where all these components work together. So you ask, well, now how do they interact with each other? So in the case of the brain, that's how they are connected, which neurons talk to whom and how, right? So that's the second part of the puzzle. And the third one is what they do as part of their function. So in the living brain, in a awake animal, what do they do? For the case of neurons, you can say what their patterns of activity are in the living brain. So the idea was, with this expansion of the Allen Institute, to characterize all three aspects of it systematically and put it together, because it was clear already back then. Of course, it was clear many decades ago for anyone studying the brain that you cannot just write down a single equation like a second Newton's law and then everything just solve everything that way. So you probably have to run a simulation. Right. So the idea was then we collect this data, we collect that systematically for all these three aspects that I mentioned, and then let's try to put it together in a simulation. Right. And a simulation that we then can use to see how well we understand the system. Can we reproduce what we observe experimentally? Let's say, does the structure that we put in the simulation give rise to the activity that we see?
Physiological activity, for example.
Yes.
Yeah.
And then on the other hand, if we have it working, can we learn something from those simulations? We can do perturbations in silico that can be difficult to do in vivo. We can learn something about mechanisms at play and so on and so forth. So that was, that was before I came. That was a part of the vision. I think Christoph Koch was really instrumental for this vision. Of course, he used to be at Caltech. He worked in computational neuroscience and brain modeling for a long time. So it was natural for him to promote that. And then I was one of the few, young assistant investigators who were hired to start carrying out this mission. And then between a few of us, I think the idea was to do it at different levels of resolution. So there are different approaches, of course, in neuroscience. There are more theoretical approaches. There are more detailed biophysical approaches. And so we wanted to do this at a few different levels of resolution. And I guess given my background in this very detailed molecular simulations, it seemed like I'd be a good candidate for doing detailed cellular level simulation.
So you got the most complex end of the stick.
I'm not complaining. I think that's what
I agree. No, so because the model you sort of ended up, this was a paper then was published in, I guess, Neuron Bille et al. In 2020, which is of course linked to in the whole page for this episode. But it sort of is, I guess it's a model for the primary visual cortex in mouse. I think I read 230,000 neurons. Within like a diameter of say like 1.8 millimeter, like a little sort of like a cylinder, almost like roughly corresponding to the size of the mouse primary visual cortex. And then there were two variants of it. One had like this core, like 50,000 neurons in the core, like I think it was like 0.4 millimeter diameter or something, or radius
0.4 millimeter radius, yes.
Millimeter radius, exactly. So that was like the center where we had like biophysically detailed neurons, and then we were surrounded by this annulus of point neurons, but of this sort of like a variation of this leaky integrating fire type. But that was like one variation, and the second was purely sort of like using this leaky integrating fire neuron types. But in some sense, you made like even though both were sort of, I would say, complicated models, but one were even more complicated than the other, at least because it had this biophysically detailed, like multi-compartmental neurons in there at the
Yes.
same time. And they had so one aspect was that I think you had a realistic neuronal density, right?
Hmm.
Yeah.
Yes.
So that was considered obviously something that you wanted to keep. So it's the right number of neurons per whatever cubic millimeter or whatever, or density per volume inside the neuron, inside the cortex, inside the sorry network model.
Right. Well, yeah. So I think, just to emphasize again, the whole idea of this exercise was to integrate the data that we were obtaining as the whole institute, right? There are many different models that people have built over the years, including models of the visual cortex. Used to be mostly in cats or monkeys, and more recently in the mouse. But uh, and there are different levels of resolution that can be useful for different questions. What we wanted to do is to stay as close as possible to the data that were available from other sources and that we were obtaining experimentally at the institute. And so, yeah, density of cells seemed like one of the major such parameters, where we know there are multiple cell types. We know approximately, at least, how they are distributed in that part of the cortex, and we want to capture that because it's probably there for a reason. So we want to capture the real thing.
Exactly. So because then I think there was one early paper I think coming in 2016 or something from Allen and Teeter et al.
Hmm.
Where which was on how to get neuron models from these experimental data. So because you did all these like patch electrode recordings in at Allen,
Right.
and then you extracted this something called like generalized leak integrated fire models from these
Hmm.
data. So so what are these GLIF models, generalized leak integrated fire models?
All right. So let me just also give you a little bit of a background here,
Yeah.
just for better understanding. Uh, so this all has been collaborative work, right? So we were talking about how the Allen Institute is sort of special. Our motto is big science, team science, and open science. And so the team science part is really important because we are doing big projects, and we have to, many groups have to work together. And so that's true also not only for us computational people using experimental data of our colleagues, which is crucial, but also it's true for the computational groups working together. And so for this modeling effort that we are talking about here, there were multiple modeling groups working together. In particular, I want to emphasize, so on the one hand, my group, on the other hand, the group of Stefan Michalas. And so for my group, the task was more to do research with biophysically detailed models of neurons with Hodgkin-Huxley dynamics and with capturing their dendritic morphologies. And for Stefan and his team, they were focusing more on these point neuron approaches. And so when we are talking about the GLIF, generalized stick integrate and fire models, this is actually work of Stefan and his group. The paper that you are referring to, Gaute, that was published, it was in 2018, I believe, in Nature Communications. We had two papers in that same issue of the journal. So one was Teeter et al. About the generalized leak, integrate, and fire models. And the other was from my group with the first author, Nathan Gowans et al., about building models of the Hodgkin-Huxley biophysically
Wow.
detailed cells. So so I'm happy to tell you about the GLIF neurons, but I just have to acknowledge right away
Sure.
that
Yeah,
this is not
absolutely.
my work, and this
No,
is the work
but
of my colleagues.
please tell us about the
Yeah,
GLIF models first, what they are as well.
yeah, yeah. But
Mhm.
the idea is that just a regular leaky integrate and fire cell is probably the simplest possible model of a neuron that you can think of, where there is some kind of capacitance and conductance associated with the neuron. it can accumulate charge, and also it can leak charge, right? And then the voltage that represents the membrane, the difference of potential across the membrane at the soma of the neuron. Once it reaches certain threshold, the neuron fires a spike. It's just an event. We say, okay, there is a spike, and then after that, there is a period of silence, usually a couple milliseconds, right, where the neurons is not allowed to fire it, and its potential can decay some further. And so this is the simplest model. This model is already quite powerful, but it has certain deficiencies. So one key deficiency, for example, is that those models don't exhibit adaptation. So in real neurons, if you take a cell in a slice of a brain, a neuron, right, and you do a patch clamp experiment on it, and you inject current into the cell body of that cell. If you inject enough current, the cell fires action potentials. Now, if you keep that current constant, the cell might fire action potentials with certain frequency. But what is typically observed, the adaptation of the spiking, is that frequency might go down, right? So the spiking might become less and less frequent, even if you keep the level of the current the same. And so that's, for example, something that typical or basic leak integrate and fire model won't capture. You inject certain amount of current into that cell, it will keep firing action potentials with certain frequency, right? So now there is a whole family of methods developed by many people. So, for example, Wolfram Gerstner is one of them, and then people like Izhikevich, say about 20 years ago, developed various approaches to model a variety of behaviors of cells with such simple spiking models or point models of neurons that capture things like adaptation and other properties using different mathematical tricks. And so, generalized leak integrate and fire model, in this case that we are talking about, developed by Stefan Michalis and his group, they introduce additional mechanisms. There are actually a few different levels of this model that has more and more mechanisms that allow you to capture different features of electrophysiology of the cells that most basic leak integrate and fire model doesn't capture. The one of the typical approaches there is that there are after spike currents, right? So after the spike happens in this point model, there are additional currents that are produced by that model with certain time dynamics. There also can be a thing like the threshold at which the cell spikes is not constant. There is also some dynamics associated with it. And so once you have that, this is just a more versatile model. It's still computationally very, very efficient, but it's much more versatile, and it can capture certain things, like, for example, this adaptation that we've been talking about. And because of that, those models, even though they're still computationally quite efficient, they can be much more powerful in describing what the different cell types do. Because a lot of neurons, they differ in the patterns of electrophysiological activity, like, for example, how much they adapt once you inject current.
And then, I think in this teacher paper, this was used. Well, they had this procedure algorithm for fitting these GLIF models to this, I guess, patch electrodes recordings. But they had from many different types of many of these the types of cells. They had these different transgenic lines,
Yes.
right? So what was that all about?
Well, that was exactly one of those three major things that I mentioned, because when we are trying to understand the system or build a model of it, we are thinking about the components, interactions between the components, and the function or activity of the components. And so here it's about understanding the components, which are the different cell types. So we are
So
focusing
these are
on
like the different, like for example, like these different types of interneurons, For example, VIP and PV and what they called like these
For
different.
example, yeah, so we are focusing on the mouse visual cortex, right? That's our
Mhm.
model system, and now, so the most recent description of cell types by the Allen Institute, according to their transcriptomic properties,
Mhm.
shows that if you look at mouse primary visual cortex just by itself, V1, there is maybe about 100 different types of
Okay.
neurons just
Oh.
there. That's a lot.
Yeah.
One can argue whether all of those 100 cell types are meaningful or how different they are and so on. I think a lot depends on exactly how you what you call a type. But from a long time ago, of course, like basically since the time of Cajal, more than like 150 years ago, it was clear that there are cells that they just look different. Their morphologies are different, even in the same part of the cortex.
Mhm.
And so the most major division is the excitatory versus inhibitory neurons, right? And there
Mhm.
is something like 80 to 90% of them are excitatory and others are inhibitory. But then once you start looking, both of those groups have some diversity within them. And so if we take a cortex specifically, right, the cortex is a layered structure. So
Mhm.
there are multiple layers, layer 1, layer 2, 3, layer 4, layer 5, layer 6 in mouse V1. And then if you look at a particular layer of the cortex, the excitatory cells, they are somewhat different. Uh, the most salient feature being that if you are, so there is this layered structure, if you are in, say, layer 2, 3, the neurons usually have this apical dendrites that point upwards, right? And they go typically all the way to the surface of the cortex. And so if you are a neuron in layer 2, 3, somewhere here, you have a very short apical dendrite. But
Mhm.
if you are in layer 5 and you're an excitatory cell, now you have a very long apical dendrite, right? So right there, you have just difference in size and the amount of branching and so on.
Mhm.
And those cells also show somewhat different electrophysiological properties.
Mhm.
And then on the other hand, you have diversity of inhibitory cells also, and there are these major subclasses that by now people have studied quite a bit, which you just mentioned, like VIP, SST, parvalbumin, or PV
Mhm.
cells, and so on.
Yeah.
And so the idea was, we our colleagues here at the Allen Institute, they characterized this diversity using electrophysiological and morphological methods in slice, and they use these markers like PV, VIP, SST to tell us at least approximately which neuron type they are working on. So again, we know that there are multiple types within those classes. So like SST, for example, we think there are more than 20 types of SST neurons. But let's start at least with these, the level of diversity,
Yeah.
like SST, VIP, PV, and some types of excitatory ones. Characterize their electrophysiology and then fit the models to match those
Mhm.
characteristics for the different cell types.
Mhm. So then you got this library or database of different neural models, I guess for
Yes.
like 16, 17 different types of neurons, right? For because I had like each layer maybe had like one excitatory and three variation of inhibitory, and then you had several layers. So it added up to 16 or 17 or something like that, right?
Right. So this is now a sort of an approximation or decision that we
Mm.
made when we were building the network model.
Yeah.
For this, what we were just talking about, for fitting the single cell models, uh, we simply fit them for individual cells,
Mm.
right? And so Stefan and his group did that for the GLIF models, so for point neuron representation, and my group did it for the biophysically detailed Hodgkin-Huxley based model.
Mm.
Um, in Stefan's case, they didn't need the morphology; they only need the electrophysiological signature of that cell. They get a model of that particular cell.
Mm.
In our case, we need morphology, and so for the cells for which we have both morphology and electrophysiology, we build a Hodgkin-Huxley model with full dendritic morphology.
Yeah.
And
So how
now we
did you?
have a
Yeah.
library.
Yeah, but how did you? So when you fit this Hodgkin Huxley style, this neural models with a like a multi compartment morphology or like a dendritic morphology. So then you had like you typically had like sodium and potassium conductances in the soma to make it firing? Did you have also, do you have some active conductances, or it was mainly passive conductances, passive dendrites, or passive conductances in the dendrites?
Well, so we tried a few different approaches, and there are two major constraints there. One constraint is that the more active conductances you use in more compartments of your morphologically detailed neuron, the more computationally expensive it becomes. Right? So the typical neuron model that we are working with would have hundreds of compartments, out of which only one or a few are somatic ones, right? And so if we put all the active conductances there, the computational expense is substantial, but it's not too bad. If you put active conductances, non-linearities, everywhere in your hundreds of compartments, it becomes much more computationally expensive. So that's one constraint. The other constraint, and that's the most important one, is actually availability of the data, because to be able to constrain your conductances in the dendrites, you need to know something about what's going on there. And there have been studies, and there are studies even more recently, which probe that. And there's definitely a lot of interest in modeling work to be done there, and people have done such work. But in our pipelined approach with these hundreds and thousands of cells that were characterized at the Allen Institute, we had almost none of the dendritic recordings. So all of these recordings were done at the soma. Still extremely important. It's very, there's a lot of diversity there and a lot of things to learn. But essentially, we didn't have information about dendrites. And therefore, we decided, for computational reasons and for this data availability reasons, to focus on the soma. And there at the soma, yes, we used about 10 different conductances, a few different sodium, and a whole bunch of different potassium conductances, based on what we know from the literature, some calcium conductances too, and then used evolutionary optimization algorithm to optimize these parameters to match the data.
So now you have, so all these models now, both the GLIF type and the biophysically detailed ones, are now available in databases, and it's like so everybody can actually use, whether they want to build circuits or not, they can all go in and look at the models and look at the data it was trained from and tuned on and everything. So it's quite a resource.
Thank you. Yeah, it was, for me, was one of the first experiences with this open science in neuroscience approaches. Was very, very exciting. I also want to say, a couple quick things. We were talking about the models with the active conductances in the dendrites. I should mention that there was also a group back then at the Allen Institute of Costas Anastasiou, who later moved to Los Angeles. He's at Cedars-Sinai now. And so his group was doing more of the work with the active conductances in the dendrites, and they actually made some of those models publicly available. Again, there are questions about availability of data for those, but one can do some modeling there, and those models are available as well. And then secondly, it's I just cannot emphasize enough how much of a group effort it was, right? So there was contributions from Stefan's team, from my team, and computational aspects, but we all, first of all, we stood on the shoulders of the giants in terms of the data, right? There's all these wonderful people at the Allen Institute who obtained all this data systematically under standardized protocols, very painstakingly. And these are difficult experiments to do it over and over again for thousands of cells. And a lot of those people are, TAs, RAs. We are not in the university. RAs, research associates.
Okay.
Right? Who became really proficient in doing this. And we also stood on the shoulder of giants in terms of people who have done that type of work before, right? We did something new in terms of scale, but we reused a lot of the methods that people before us published in terms of optimizing models, which usually they would do for one or two cells, and we did it for hundreds. But we really reused a lot of what had been produced in the field by people like Idan Segev and actually people in the Blue Brain project and others.
So how many cells? I mean, it's like a few hundred cells you have now, or we have models in available through the Allen database. I mean, at least we talk about hundreds, aren't we?
Yeah, I lost count at some point. I'm not sure exactly, but I think it's a good number of hundreds. So for the GLIF model, I remember at some point that the number was like 700. It's probably more than that now. For the biophysically detailed model, I think so mostly it's mouse models, but also some human. And I believe we have something a couple hundred, probably 200 or more. And again, it's been a while, so we might have some more now.
Yeah, but it's really because being a computational neuroscientist myself, right? And relying using other people's neuron models. Back in the days when it started in the 90s and so on, I mean, there were like a handful of models you could use. I mean, like a one or two, and they were beautiful, but they were sort of like used all over and over again.
Yes.
Uh, and so there was like this, I would say, like almost like a big phase transition, as it was in physics. But now we not only have one model, we have like when you go to like the Allen database, you have like hundreds of models, and I think also with the Blue Brain and database also have hundreds of models, and there are of course also models or databases of morphology, like the neuromorpho.
Hmm.
So there we have a lot of, but many access to many morphologies. But I mean, from a modeler's perspective, it's really nice that we don't have only have like a handful of anecdotal cells. We have like many neural models to explore and to and to play with. So now we have the neural models. So that's a starting point. But then you have to start putting them together into networks. And then, of course, one thing you could do then is sort of well, of course, you have this library of neurons, and you know roughly the number of well the densities of like
Hmm.
the number densities of these different neuron types in the different layers. But
Yep.
then you have to start connecting them, and so how did you go about doing that?
Yeah. So that starts from the question that you asked a little bit ago, which is so we decided to set up certain number of cell types in the model for each layer, right?
Mm,
And the decision was based on the availability of the data
mm,
already back then, which was like 2017, 2018. We had the data based on transcriptomics to tell that there is probably about 100 different cell types in the mouse visual cortex.
just one sentence of what transcriptomics is, so that people.
Right. Yeah.
Yeah.
So each cell in our body has the same DNA, right? Which contains a number of genes, thousands of genes that make us who we are. But different cell types express different genes.
Mm,
So in some cell types, that gene that you have is silent, and in other cell types, it's being produced and turned into protein, and it carries out its function in that cell. And I think this is a very simplistic description. It's like
yeah.
some probably more complex than that, but sort of zero level approximation. That's what it is. And so
Mm,
that makes cell types who they are. Which genes are expressed
mm,
determines what the cell does.
mm,
And so there are modern methods that allow us for individual cells to profile which genes are expressed
mm,
quite precisely. And that's a wonderful tool to try to understand diversity of the cells, because then you can profile thousands of those cells. For each of them, that's transcriptome, so which genes are expressed in that particular cell, and then you can use that to classify cells. And that's actually the foundation of the classification of the cell types that the Allen Institute and other people are doing these days so successfully.
mm.
So now
Okay.
this transcriptomics tells us that there is 100 cell types in V1, right? But we didn't and still don't have the data to operate at that level of resolution for really anything that we wanted to put in the model, like properties of cells, electrophysiological properties of cells, or connectivity between them, especially for connectivity, because then if we have 100 cell types, to characterize connectivity between 100 cell types, you need 100 times, to the power of 2, 10,000 matrix elements that tell you what the probability of connection is between those cell types, and
But
so on.
that you don't get from the transcriptome, right? That's something you have to
Oh,
get
no.
from. No, that you have to get from like really paired
No,
recordings or
exactly.
something. Yeah.
Exactly. That's my point. From the
Yeah.
transcriptome, you sort of you get a picture of what is there, right? Like you if you think about a city from the transcriptome, you might get a picture of which different types of people there are in the
Yeah.
city, like approximately, or how they are dressed up, let's say, right? Some wear skirts, some wear overalls, but you don't know their functional properties. You might guess, but you don't really know what kind of job they do. You don't know what they tell to each other. So for that, you need different methods. And so connectivity, for example, there are methods like multi-patch synaptic physiology, where you patch cells in slice, multiple ones, and you excite one cell and you listen whether the other cells are connected to it or not. Or electron microscopy, which gives you a wonderful resolution, but it's very, very hard computation, very, very hard problem to make sure that you know which cells are connected to which, even when you have the data.
Yeah.
And so when we set out to build this model, we decided, okay, given the level of resolution that we have for the most data, we are not going to use 100 cell types. Instead, we are going to use a few cell classes per layer. So we ended up with 1 excitatory and 3 inhibitory cell classes in each layer. So PV, SST, and VIP inhibitory. Then you have layer 2, 3, 4, 5, and 6. So 4 of them times 4 cell classes, that's 16. And then there is also layer 1, for which we use just 1 inhibitory class. We know there are many more types there, but again, as an approximation. So 17 cell classes in total. And then for connectivity, we had some information back then from the Allen Institute, but it wasn't, it was only starting the data collection. And so we had to rely a lot on the literature. And that was quite an experience to go through that literature.
Yeah.
Because, people have done amazing work in that area, right? Like each experiment is really, it's like a whole PhD can be essentially, collection of one data set, where for particular cell type in a particular layer, a graduate student painstakingly over years collected, I don't know, like 50 pairs or 100 pairs, out of which, 10 were actually connected. I might be exaggerating, but the numbers are of that order of magnitude, right? And then you get your probability of connection, and you get the physiology of that connection. You publish a paper, right? And then as the modelers, we have to go through all these papers. There are probably hundreds, if not thousands of them, and carefully look through the data, through the experimental methods, because people have been using animals of different ages and different physiological solutions and different temperatures and all kinds of different things, statistical methods. So it's like Christoph used to like to say, neuroscientists are more likely to use each other toothbrush than to use each other methods.
Yeah.
Which I think it's not quite true anymore.
No.
But it's wasn't far from true some time ago.
Yeah. I guess another type of data, I just remember that you have, like, if you sort of fill these axons and dendrites, you can sort of see a little bit that some axons from neurons in, say, layer 5, they project to layer 1 and so on. So there are like information from this, I guess, light microscopy also, right? Which you can sort of also add to it, I guess.
Yes. So that's true. And so basically, what some people at least call Peter's rule, right? You get your morphology of the neuron with the dendritic morphology and axonal morphology, And let's say you get it for cell type A and for cell type B, and then you can look, given cell type A and cell type B, you have the dendritic morphology here and axonal morphology from this cell, and you look at the overlap then, and where they overlap, you can sort of deduce the probability of connection based on that. So we tried to avoid using that for a few reasons. One reason is that for vast majority of cell types, there was simply not enough information in terms of axonal and dendritic clouds. And we felt like, one can use methods like sort of generating them based on the little amount of data that is available. But we weren't sure how reliable it's going to be. And then secondly, already back then, it was pretty clear that there are notable exceptions to the Peters rule, where you just take the overlap and you get approximately the right probability of connection, because there were cases where it was shown that it's actually quite different, quite more biased than just the overlap. And now, I don't know if this question if we can say it's fully resolved, but we see that we now have much more connectivity information, and it seems pretty clear that in a lot of cases it's much more specific than just the overlap.
So that the Peters rule may not be applicable in many cases.
Yeah, I think one has to be really careful with such statements, right? I don't want to say it's wrong or it doesn't work. There might be some relatively simple modifications that one could introduce and then capture the observation. It's just something that we weren't certain about and wanted to be careful about.
Okay, so now you almost have a model, right? So at least you sort of have this. Okay, so you have these neural models that you used, and you hook them up using information from the literature and in-house recordings about whether they are connected, different neuron types, and the probability of being connected, and then maybe also some distribution of how strong the connections are in terms of how big the effects of synaptic inputs are on the, I guess, EPSPs, the potential in the soma on the receiving cell. And then you want to sort of another, of course, one way to test it and tune it is to compare with experimental data. And I guess that's one that was also done. I mean, this really beautiful and comprehensive work done. I guess the first paper by was, again, a group effort, I'm sure, where I think the first author was Josh Siegler, right?
Yes.
Where he did like this very standardized electrophysiology measurements of using this neuropixel probe, also developed at Allen Institute in collaboration with others. So tell about this kind of the physiological data that was recorded.
Right. Yeah. So that's the key, right? We've been talking a few times now about this three major aspects of how you characterize the system or how you build a model, or what I like to call three pillars: the structure, the connectivity, and the activity or function. And so this is the third pillar. Now we want to know what our cells, what our neurons do in the living, breathing animal. In terms of their activity. And so for that, multiple methods exist. There is the calcium imaging, for example, which people at the Allen Institute also used, and that was a useful data set for us as well. And then there is the electrophysiology, which, in a nutshell...
calcium imaging, that's optophysiology, right? So
yes, it's a microscopy, fluorescent microscopy, yes.
Yeah, where you measure the calcium dynamics and get spikes and activity from that. Okay, and then you have electrophysiology.
Yes, and you have electrophysiology, which essentially is, you insert a thin probe in the brain. You hope not to damage anything by doing that. The probe has an electrode on it, or in the case of modern probes, it's many, many electrodes. And then each electrode measures the electric field or the electric potential that's created by the activity of all the neurons. Well, you have been working on it for a very long time and have a lot of
Absolutely.
papers, and more recently a beautiful book that really goes into the
Absolutely. Yeah,
details of
coming
it.
out this summer. So
Yes,
electric brain signals. Yes,
right.
indeed.
And so essentially what happens is that the electrodes can then pick up the activity. Well, they can pick up the activity of the population of neurons, sort of in the low frequency domain, but also at a high frequency level, they pick up activity of individual neurons that probably are very close to that electrode, and then you can capture the spikes that those neurons produce, right?
Mhm.
And so this is something again that people have been doing for decades, but recently there had been a development where, in particular, Allen Institute was playing a big part. Development of these wonderful neuropixels probes, which have very high density of electrodes, really nice properties. They are versatile and powerful. And so with that, our colleagues at the institute, Josh Siegel and others, were able to record. I'd say about 1,000 neurons in a single mouse in a single experiment
Mm.
simultaneously.
Mm.
That's using about six neuropixels probes,
Mm.
and so they go through different visual areas and also subcortical areas. If we are talking about a single cortical area like V1, in a typical experiment, they were getting about 100 neurons recorded.
Mm. Yeah.
And then that was done in over 50, about 60 mice, well, at least in the first data set. The data set was again made publicly available before
Mm.
any publication of ours. And that was an amazing resource for us because now you have hundreds, actually thousands of neurons for which you know their responses in the actual mice
Mm.
um,
And
with high
also,
precision.
for a set of visual stimuli, right? I was like a fixed protocol of gratings and movies and images and all
Yeah.
kinds of things. Yeah.
Exactly, yeah. So because this is the visual cortex, we want to say something about its function, and well, the function is probably related to the visual
Mm.
processing.
Yeah.
So in the experiments, things like drifting gratings, so natural images and natural movies were shown to mice, multiple sessions. We record
Mm.
activity. So now we have a benchmark. We know what those neurons do in response
Mm.
to this visual stimuli, and the question is, can we reproduce that in the model?
Yeah. And I think, I mean, this resource, I think it was like 50, 60, 70 mice or something, the same age. Everything should be as reproducible as possible. So it's a fantastic resource, regardless whether, even for those who are not interested in modeling, just
Yes.
to see what is the natural variability. How different are two mice really, right? Because typically it's been
Yeah.
difficult to assess that, right? Because it's been difficult to record this, well, record from many mice in like of the same type and in the same situation and so on.
Yes.
So that is really cool just by itself. But then you use this data to sort of to try to tune connectivity parameters, right, in your model.
Uh, yeah, sort of. So that's actually a very interesting point about what we did with the model.
Mm.
This is a bottom-up model building, right? This is like
Mm.
you have the components, you know how they interact with each other. You put it all together, and in the ideal world, if you have all the data
Mm.
and you put it all together correctly, you expect that you turn it on and start producing the activity that you
Yeah,
observe in vivo.
if it was perfect, right? If it was really, yeah.
Yes.
Yeah, yeah.
The reality, of course, is that there are a lot and a lot of caveats and unknowns. We have some data, but we don't have all the data. V1 does not live in isolation. Probably 60% of synapses, if not more, comes from outside of V1.
Mm.
Uh, there are a lot of things that can go wrong.
But even I just forgot. I mean, there's one thing missing in the model, right? You need, if you want to compare with results to, I think you'd compare a lot to like drifting ratings. You also need a model for what comes into V1 from thalamus.
Right.
So that's a separate model, right? Which you had also to construct, right? So how did you do that?
Yes, yes, that's a very good point. Because we wanted to be able to connect to compare apples to apples on the
Mm,
model of V1 with this recording of V1 in response to visual stimuli,
mm,
we wanted to provide such inputs into our V1 model. And as everything in the brain, it's very complex, and we
mm,
know relatively little about how it actually works. So we know that photons come to the eye, then in the
mm,
retina, they are converted to electric signals through actually multiple layers of multiple cell types. The signals then travel to the thalamus, to the
mm,
lateral geniculate nucleus, or LGN of the thalamus, and from there to V1. Then, of course, there are multiple parallel pathways or other pathways that also convey visual information that eventually ends up to V1. So this is just one major pathway.
mm,
So still, we tried to model it. We didn't model it explicitly. We modeled it as a layer of filters. So very similar to what people do in like in computer vision or in artificial neural nets working with images or movies, right? We have a layer of filters that have some receptive fields in this visual space and have some kind of temporal properties. Then we show a movie to these filters, and those filters convert those movies
mm,
to the output, to a firing
mm,
rate output, which we then can convert to spikes.
mm,
And then, so actually, a lot of this work, again, I must say, this was collaboration. A lot of this work was spearheaded by Stefan Michalas and his group. Then my group worked a lot on like how we take these filters and connect them to V1 cells.
mm,
That's really not very well understood. How LGN cells with their different properties connect to V1 cells. There was some data coming out in papers, and we integrated that. So in the end, it's there are a lot of approximations there, and we know that some things are not quite right.
mm,
But the great thing about it is that now we can send in an arbitrary movie, whatever visual stimulus we want. We can pass it through that layer of filters. It will create activity that's sort of like the one in the thalamus, and it will then travel to the V1 cells and excite our V1 cells. And that way we can use exactly the same visual stimuli as our experimental colleagues
mm.
have been using.
Yeah, this is what we call, I guess, the idea of having this digital twin. That is what people call it these days, right? They try to make something that in silicon that mimics the real thing. I guess, I mean, this input that comes from the thalamus, which is sort of like it's like filter model or descriptive model, or has
Yeah.
a statistical model, has a long tradition in visual neuroscience, just
Yes.
beyond sort of computer vision. I mean, with this difference of Gaussian's model
Hmm.
and this Gabor filter models for V1 and
Right.
so on. But I guess it's this combination of that you use a descriptive model as input to provide the input to this mechanistic V1 model, where you sort of try to make a model based on neurons and connections.
Yes. Yes. And yeah, for us, it was really important to be able to compare to experiments in a more more precise way than just say, well, there's that much firing here, that much firing there on average, right?
Yeah.
And I think there is another really key important aspect of it, which when you do this, you really have to think carefully about things like, where are your inputs from the different thalamic cells coming, how they organized. And in the end, it's all about this, retinotopy and the relationship between your physical arrangement and structure of the network
Mm,
and its function. And it's non-trivial, but also it's ultimately that's what subserves the function of this
mm,
network, right?
mm,
So because we added this layer of complexity, we actually now have a chance of simulating some interesting and complex computation in the
mm,
model, right? Because
mm,
it's not just there is a lot of important work that one can do with a network that just receives some kind of barrage of spikes, and then you can ask how sort of population level activity develops and so on. But with such very fine and structured input, we can actually look at very specific computation that the network does, on a particular types of visual stimuli, and that's, it's a can be powerful. I
Yeah,
hope.
yeah. So, so now you have this model, and then I guess, but then you need to to run it, and you need they need to be able to have like a computer language, programming language. So you even made your own programming language to be able to simulate this, right? This B, or at least this BMTK. Well,
Right.
yeah.
Yeah. So it's a API, right? A user interface, a Python-based software package
Mm,
to run simulations. And again, here, like with everything else, I like to emphasize this again and again. We stand on the
mm,
shoulders of giants. There
mm,
had been very powerful software tools in neuroscience long before I joined, like for example, Neuron or
mm,
Nest, the programs that allow people to run simulations, to build sophisticated model, to run them efficiently in parallel. What we found out when we were working on our network models of V1 is that, yes, these tools exist, but you still have to write a lot of code. For example, in Neuron, there was just nothing that we could take off the shelf. We had to write a lot of code. We had to learn sort of the insights of how the neuron works, and write the code to build our model and to run the simulation of this model in parallel. The neuron gives us the tool to do so, but you need to master the tool.
mm,
And so when we did that, we wrote a lot of code, which we then figured out, well, we can reuse it for other network simulations, and we can share it with the community. And so that gave rise to BMTK, which it's an abbreviation for Brain Modeling Toolkit, which is our Python package, which we share with everyone. Uh, it just provides useful functions in Python to build the models and then to run simulations either with Neuron or
mm,
with Nest or with couple other things that we developed at different levels of resolution.
mm.
So
Or even run the model that you published in
yes.
2020, right? Yeah.
Yes.
Yeah.
And another aspect of it is that also because these models are very, very heterogeneous and sophisticated, we realized that we need some kind of standard file format to keep all that
Mm,
information about the model.
mm,
And then, we started developing something of our own. Then we realized that other people have similar needs. And
mm,
we joined forces first with the Blue Brain, people in the Blue Brain project, and then with a few other groups like Porin Glissan and Salva Dura Bernal and Andrew Davison. And so we published a paper about this file format called Sonata,
mm,
which allows us to store all the information about models.
mm,
So that's what we use. We have a model in Sonata format. One can take our tool BMTK to run that model and just take the Sonata files and runs a simulation for you. Uh, or you can actually use other tools that understand Sonata. And so, for example, we were recently working with someone you know very well, Hans Egerhard Plesser,
sure. A good
right? With
colleague at NMBU. Mm,
yes. So with this Nest group, and now they implemented native support of Sonata in Nest. So now for point neuron simulation, you don't need BMTK. You can just
mm,
take Nest, take our model in Sonata format for point neuron version of it, and you can run it in Nest natively.
mm. Cool. So and then in order to run your models, I guess I mean you had two versions of the model. One, as I mentioned, one with only glyph, 230,000 glyph neurons, and one with whatever 50,000 biophysical detail and whatever 180,000 glyph neurons. So
Yep.
how did you run them? Do you have super duper computers at Allen where you where you run these things, or?
Yes. So we have a really good cluster at the Allen Institute. I guess one could call it supercomputer, although compared to the huge National Science Foundation Supercomputer Center or other
Yeah.
huge supercomputers that you can find around the world, it's not that huge. Um, but yes, we mostly ran it in our local facility. The point neuron model really is not that compute expensive. You can run it even on a single CPU.
Yeah, no, that I know actually. It's we have had like master students working on it on a normal like office computer.
Yeah.
So that's doable. Mhm.
Yeah, and you can get maybe 1 second simulated within a few minutes.
Yeah.
So it's not real time, but we're talking single CPU with 230,000 neurons, and it can
Mhm.
parallelize on multiple CPUs. And then for the biophysically detailed model, it's more like you need something like say 500 CPUs and 500 cores, right? So
Yeah.
modern CPUs usually come with like 32 or 64 or more cores.
Mm.
So let's say you need 500 such cores to
Mm.
run a simulation with a reasonable throughput, and then you get something like one simulated second in 1 hour,
Yeah.
which is much slower. But again, as you know, and your group had been running such simulations,
Yeah, we have
it's
used a
doable.
lot on. It's doable, and we have been using. I'm lucky to use supercomputers in Jülich, Eugene in Jülich in Germany. So that has been very useful for us. But then when you have this this model, right? I mean, it's how do you? I mean, I think the traditional way that we were trained in physics, maybe that we when we sort of had like we made this. We don't want to fit the model to data. But typically,
Mhm.
the model was quite small. Maybe had like one or two parameters. Was it like in was sort of like sort of low dimensional, whatever, and
Yes.
and then so that you could sort of fit it to data and maybe get the unique parameter set that was like the best fit where the deviation between the model predictions and say experiment was
Yes.
the smallest. That was sort of the typical way to do it. But this is not how we can think about these models, right? With there's so many parameters. Or how do you think about this issue of finding identifying correct parameters?
Yeah, that's the key question.
Mm.
And so, Gaute, you have a really nice review paper about it in Neuron 2019, I believe, right? The
Yeah, that's
multi-purpose modeling.
yeah, yeah, that's true. So where we try to, yeah, that's true. Yeah, but that's sort of more on the sort of why we would like to. Of course, we have also co-organized our workshops on multi-purpose modeling,
Yeah.
where one aspect is that and you would like maybe to develop a model that can sort of predict many things at the same time. Uh, and but it's also a question of sort of how do you. I mean, one big problem here, I think, is sort of or how should we think about. I mean, it's not about finding one unique parameter set. And that is sort of
Yeah.
it's more about finding like a class, different classes or sets of parameters which are
Right.
compatible with data, right? So we have to learn to think differently about these things.
Yeah, yeah, exactly. And so why I'm bringing this up is because your paper argues very nicely and eloquently that we should be thinking about multi-purpose models, right? That the models, and that's definitely how we approach this. The models that we build, the idea is not to ask a particular type of question. The idea is to create a computational platform for discovery, right? The
Mm.
hope is that we will put in all that complexity there that we don't even know what is it for. We just say, well, this is what we observe in the world. There are all these cell types, there are interactions between them. Let's put it all together and hope that we get it right.
Yeah.
And then you can use it to answer a lot of different questions. You can ask questions about disease, you can ask questions about just basic functions of the brain, you can ask questions about spikes or electric field or
Mm.
magnetic resonance imaging or whatever. But the point is that it's the same model that you use for those multiple purposes. But if that's the case, then the problem is you don't know what to optimize it for,
Mm.
right? Because if you know which question you are trying to answer, then yeah, of course you can build the cost function based on that. But if you don't, then it's sort of arbitrary. And so, okay, so then now there is this problem. So how do we approach it? We still want to build our model and do something useful with it. So what we did, we said, well, let's just try to keep it very simple. We put all this data that we can integrate, we put it into the model. We try to not touch it as much as we can, right? So whatever we put in, to the extent that we know that it's at least roughly correct, we try not to touch it. But what we will try to optimize the model for is the most simple thing that we can think of, which is to reproduce approximately the firing rates of neurons during a visual stimulus. And that's basically what we did. Now, there is another very interesting aspect to the story, which is, let's say, if you take the modern machine learning or artificial intelligence, when people optimize those sort of nets, a typical approach, as far as I know, is let's say you have your 100% of the data with which you want to work, then you maybe use 80% of this data as your training set, right? That's what you train your model for or on. And then the remaining 20% of your data is the test set, right? So you trained on 80%, then you test how well it works on the remaining 20%. We didn't have the luxury of doing that because our models are too computationally expensive.
Yeah.
80% of the data is just completely out of the question. So
because you're not able to run the model so many times.
Exactly. Yeah. You run 1 second in 1 hour.
Yeah, I know. Exactly.
A single session with NeuroPixels is at least an hour. It's more than an hour with one mouse.
Yeah.
So, so what we ended up doing, we used a really impoverished training set. Our training set was 1 second, 1 second of dynamics, where half of it was the gray screen, spontaneous activity, and the other half was one single drifting grating. And all we wanted to reproduce was, let's have our spontaneous activity be roughly in the ballpark of what is observed experimentally. Let's have our evoked activity to be roughly in the ballpark of what observed experimentally, right? It's just the magnitude of the firing. And let's make sure that the model is not epileptic, because what's typically happening in these highly recurrent models is that you have a little too much excitation and then everything blows up. All neurons are simultaneously active, right? And, back then we did this optimization manually. There are probably better ways of doing it.
So when you say training, you actually mean that you looked at it and tried to tune it by hand?
We tuned it by hand in the sense that we looked at, we did have specific metrics that we were looking at, right? Like we compute the firing rates, distribution of firing rates for a given cell type. We look, for example, at the median of it, right? And we compare with the experiment. So we didn't just look by eye. We did have a quantitative metric, but we didn't use a computational algorithm to adjust our parameters.
No, because then you typically have to run many times, and that's
exactly
that was beyond the
yeah. And we still had to run many times, but at least using our own brain as an optimization tool, perhaps we saved a lot of iterations.
Exactly.
And so, and then the parameters that we allowed to optimize were only the overall magnitudes of weights.
Okay.
We didn't manipulate individual weights. We would say for a given type of connection, let's say layer 4 excitatory to layer 2-3 excitatory, we can multiply all of those weights by, let's say, 1.1 or 1.2 or 1.3, and let's see, does it change what we observe? Does it bring us closer to the experimental values? And in the end, we were able to get pretty close for that one impoverished training set, right? 1 second. And then after that, we said, okay, we don't touch the model anymore. Let the parameters be what they are. And now we can do our test set, which was much, much larger than our training set, which was multiple seconds of different drifting gratings. And then we also did natural movies and look at the performance of the model.
Response, yeah. No, this is quite quite a challenge. So So how has sort of your how has the model been used? It's been out since 2020, and it's been it's available for many. I mean, for everyone, and it has been used a bit around, hasn't it?
Yes, it has. I'm happy to see that it looks like it was well received.
Mm.
So yeah, on the one hand, there are multiple users, and there are a few major types of users where some of it is through collaborations, like you and us, of course, collaborated and recently
Mm.
published a really nice paper in eLife about the local field potential and current source density using
Mm.
basically this model with some modifications, right, and relying on NeuraPixels data.
Mm.
So there are examples like that. We have a couple other collaborations where we're working on different aspects of using the model that way with other colleagues. But then it's also very nice that other people just come and take the model. They take some components of this model and to use it in their own modeling work. Sometimes they take the whole model and run some simulations with it. So far I'm aware of about 15, maybe a few more different preprints and papers that are already published,
Yeah.
that are using this model for those external use, not counting our own work or collaborations.
Is that mainly the glyph version, which is more tractable?
It is absolutely yes,
Yeah.
because the glyph version is essentially thousands of times faster and computationally
Yeah.
more efficient. Vast
Mm.
majority of users
So
are
why
using
this?
it.
I mean, I guess it could maybe be optimized this biophysically. I don't know if there's like a factor 10,000 slower or something, or
So yeah, I think it depends on how you count, but it's
yeah,
like 1,000 to maybe 5,000 times slower, something like that.
exactly. So I think it's yeah, yeah. So well, so that's it would be nice with even better computers, not necessarily to run bigger models, just be able to run the same model more
Yeah.
times. Yeah, but I'm thinking one analogy to if you sort of look at like the say AI convolutional neural networks,
Yes.
then they keep the neuron units fixed, right? And they were simple units, and all training is done with weights.
Hmm.
And it's a little bit like you also keep the neurons fixed and train with weights, right? I guess it's maybe there's our yeah. I don't know how to yeah if that sort of is the if there's some kind of deeper principle there that maybe the neurons are maybe all the learning is mainly done by also in the brains real brains are done by changing weights and not so much by changing the neuron dynamics.
Um,
Yeah. Who
I mean,
knows?
there are probably fast and slow processes, right?
Yeah,
And I mean, we know
absolutely.
that some things happen at the level of
Yeah.
neurons, but
Mm.
a lot probably happens at the level of synapses. Uh,
Mm.
I mean, personally, I want to be open-minded about all of this and the different levels of resolution that can be useful. So, if there is anything that we learn from this exercise over the years, is that point neuron models turn out to be really powerful. So
Mm.
actually, when we look at spike in dynamics, the difference between the biophysically detailed version and and the point neuron version of our models is not that big. There are
Mm.
differences that you can find for sure
Mm.
if you look, and they're not even that subtle. But for things that we compare with experiments, typically it's like overall level of firing rates, direction selectivity, orientation selectivity. The computational properties,
Mm.
the point neuron model actually can capture them pretty well. And
Mm.
if that's the case, why would you use a more computation expensive model at
Mm.
all? Of course, there are cases where it matters, especially in the work like you are doing and we have been collaborating on,
Yeah.
where it is
Predicting brain
important,
signals. Mm.
right?
Yeah.
Yeah, it's important where your synapses are. It's important how the current flows to
Mm.
get, for example, the local field potential, right?
Mm.
And that can be important for things like clinical applications. So I
Mm.
still believe doing those much more detailed models is important.
Mm.
But I think ultimately it's like you want to have an armamentarium of different tools, right? And you take the tool that's useful for the task at hand, and for many tasks, these point neuron models are just fine.
So how can a model be improved? I guess in your when in like in the Billy paper, then you mainly looked at, well, you trained it a little bit on this. Well, you didn't really train it that much, mainly for practical reasons.
Right.
So I guess that's one way to improve the model is if you say focus on this glif version and then try to fit sort of then the model to explain more of the experimental observations that you have in this like electrophysiology protocols, right, or electrophysiology data. So I guess that's one way to go, which would and one natural way and one way to move on, and then another one is if you're trying to have you tried to compare the model with like optophysiological data like calcium imaging. Is that something you've tried or?
Well, we tried as a part of that same work. The issue with optophysiological imaging is that there's always pros and cons. It has some pros relative to the electrophysiology, for example, that you can have a much better control of which cell types you are recording from. But then you are not recording spikes. What you're recording is the fluorescence of calcium indicators. So it's like there are multiple levels of removal from the actual electrophysiological signal. Well, but it's useful. It can be really revealing, and especially if you want to understand something about specific cell types, it's exciting, and we will definitely do more work on that. In terms of improving these models, I think there are a few things that are sort of happening now and are really exciting. And so one has to do with like the AI revolution that's going on seemingly everywhere. So actually, some really exciting work had been done by, for example, the lab of Wolfgang Maas, who took our model and ported it into TensorFlow and trained it using backpropagation through time to solve tasks, like behavioral tasks that actually.
So then they made it into some firing rate version or something of it, or
it's a point neuron version where it's a spike in point neuron version that still captures most of the essence of the glif models.
Okay.
Right, but it works in TensorFlow, and it can use this modern training approaches to actually train the model to do something computationally, or really whatever you want, given loss function that you can develop. So we're actually collaborating with them now to go further with that. But that's one exciting area where instead of doing this painstakingly by hand, one can try to bring in more modern techniques and compute power to really train the models properly. I think if that works, and if that works well, it's really a very, very potentially fruitful area for us to do something significant with these models. So you can play the games like people in AI and neuro AI playing, but you can do it with really rigorous biological constraints that our models bring to the table. So that's one area. The other area is we have much more data now than we used to 5 years ago. And that's something my group is working on too, where we are having the synaptic physiology data and the electron microscopy data that describe the connectivity between V1 cells. And we find out that, when we integrated data from the literature, and when we look at the data that is obtained consistently for mouse V1, for all the standardized conditions, there are actually substantial differences.
Okay, yeah.
So we don't know how it's going to affect the function.
No.
Right? It's an interesting
question. But when
question.
you say synaptic physiology, that's paired recordings, or how do we?
Yep.
Yeah, paired recordings.
Right.
Where you also look at like synaptic plasticity, or
at least the short-term synaptic plasticity.
Short-term
yes.
yeah.
Yes,
I guess the long term is much harder.
Yes.
And then you have made this electron microscopy, what's called reconstruction of a piece of mouse visual cortex, haven't you? Or are you at Allen?
Yeah, so not me, but
No, no,
people
but I mean
people at the Allen
people at
Institute,
Allen.
and
Yeah. Mhm.
my group is collaborating with
Yeah.
those groups. So yeah, that's also very exciting data set where there are actually two separate cubic millimeters of mouse V1,
Mhm.
where all the electron microscopy imaging had been obtained, and a lot of them had been reconstructed already.
Yeah.
But there are, like I said, it's computationally very difficult problem to make sure that that all your neurons are proofread and you get real connections between them. You are
Mhm.
certain about those connections. But still, we already in those data sets we have data for some thousands of neurons. We know neurons of different types connect to whom, and it's it adds very substantially to what we knew about the connectivity in
Yeah.
V1, and so
Mhm.
we can compare that to what
But
we
can you?
used to have.
But I guess you can sort of read out whether they are connected or not. But can you read out the connection strength?
It's in maybe indirectly. So there have been papers showing that connection strengths, as measured
Mm.
by PSPs, is
Yeah.
correlated with, for example, the size of the synapse, or the amount of
Yeah.
postsynaptic density.
Yeah.
It's not perfect, but even just to know who is connected to whom is already
Sure.
really valuable.
Yeah. Cool. Now we are sort of heading towards the end of the podcast, and so now we can be even more ambitious. And like, what are sort of like the prospects for? I think you already work on models for like entire mouse cortex or mouse visual cortex, or you're with like not only like 230,000 neurons like in your V1 model, but with many million neurons.
Well, yeah, we have something in the works with the model of, say, preliminary model of the whole mouse cortex, which is about 10 million neurons. It's of course
Mm.
much less constrained by data than the V1 model, but it's extremely interesting.
Mm.
We have some work where we are trying to put together multiple cortical areas, so say V1 plus some higher visual area.
Huh?
Um, yes, so I would say looking into the future. There is a lot to do, and there is a lot
Mm.
of interest in the whole brain. Ultimately, right? It's the
Mm.
whole brain that works properly together in the end. And so I would like to go to bigger
Mm.
systems, more complete systems. V1 doesn't exist in isolation. We were just
Mm.
focusing on it because of that's where most of the data were available.
Yeah.
Um, but yeah, ultimately, it would be great to go through a series of, a brain area, multiple brain areas interacting together, some substantial chunk of the brain, and then the whole brain
Mm.
all working together. There are really exciting initiatives in the field where, well, like the Allen Institute just recently published some papers, and also other people in the field published papers about characterizing all the cell types in the whole mouse brain, for example,
Yeah.
right?
Mm.
Um, then there are initiatives on resolving connectivity well beyond a single cubic millimeter of the brain, and ultimately for something like a whole mouse brain. Um, and
Mm.
models, I think, should benefit from that, and also should sort of go hand in hand with those activities, and try to help.
Mm.
And then my dream is to go to the human brain. Also, we've done a lot of work on the mouse. We have a lot of data at the Allen Institute looking at the human brain. There is a lot of interesting data for a non-human primate. So you could imagine something where there are models of multiple species brains being developed. Maybe not quite in parallel because they're very different in size and the data availability. But
Mm.
you want to have the model of a whole mouse brain, and then maybe a model of a whole monkey brain, and ultimately a model of a whole human brain.
Mm. But what do you think is holding us back? I mean, if I think about the mouse V1 model, it's very difficult to. I mean, if you to work with this well, to fit the model to experimental data, right? That's to a large amount of, well, the different kinds of experimental data, doing that efficiently, and of course they say that with like many parameters it's easy to fit. Well, you
Hmm.
can fit anything, but I mean that's not true with mechanistic models. Network models is actually quite hard to
Yes.
fit parameters. Even if you have gazillions of parameters, it's very hard to make them to fit the model to do certain things.
Yes.
So is that sort of what's holding us back? I feel that we don't really know how to, and also when the goal is not sort of to find a unique parameter set, but just like sort of find a right class of parameters, maybe, because we also know that some of these parameters change over time in real neurons, like the channel, the densities of conductances, and so on. So there's some conceptual challenge there also, isn't it? Or
Yeah, I think so. I totally agree with what you're saying, Gaute. It's the one challenge is to have, the computational infrastructure and the software and the hardware
Mhm,
to allow us to do this detailed simulations. I'm not saying necessarily by physically detailed, it can be point
mhm,
neuron, but detailed with a lot of
absolutely.
biological constraints, uh, faster, more efficiently than we do now, so that we can have not one model of V1, but hundreds of models of V1 or
Mhm,
thousands of models of V1, all with somewhat different parameter sets, so that we can explore that space of parameters, so that we can maybe develop the model not for generalized V1, but
mhm,
for individual mice,
mhm,
right? And then once you do that, you can start thinking, well, you can do it for normal mice, you can do it also for models of disease, right? And then of course we can think about not just V1 in mouse, but whole brain, so human cortical areas and such. So yeah, those types of methods and approaches you are mentioning would be very valuable, and there is some good work happening in this area, but yeah, still a lot remains to be seen and done. But in terms of what else is needed, I still remain of a very simple opinion that we are data starved. We are
mhm,
data starved field, the computation neuroscience modeling.
yeah,
We still don't have all the data we need by far,
mhm,
right? All the things that we were talking about, uh, cell types, connections between them, activity in vivo. We have amazing data now, but we are still only scratching the surface,
mhm,
right? Like
mhm,
if there is 100 cell types in V1, we don't have
yeah,
enough data for 100 cell types. We only have enough for some like higher level classification.
mhm,
If yeah, the connectivity, like we have connections for maybe some thousands of neurons,
mhm,
even in a current electron microscopy data. Activity, right? There is hundreds of thousands of cells, like roughly 370,000 in a mouse
mhm,
V1.
mhm,
Uh, in a single recording, we get 100,
yeah, yeah,
right?
mhm.
We still need more data.
Yeah. Oh, that's certainly true. But I guess I also agree that we have to work with the data we have, and
Yes,
then see where do we hit the wall,
yes.
and so on. You also told me that you also interested in the question of consciousness, and have your own little take on that, so that you have a paper out
Mm.
on. So what is now is your? I guess this is it's interesting that more and more people are getting interested in consciousness and thinking seriously about it. So what's your 3 minute story about your thoughts about consciousness? I can give you 5 minutes if that's what you need, but
Well, yeah, I'll try to be quick. Well, just on a personal note, that's something that essentially that's the reason why I came to neuroscience. I was thinking what is interesting to do with my scientific life, and I
yeah,
realized that some of the major mysteries lie in the questions about the mind and the connection
mhm.
between the mind and the body,
I
right? And
think that's
so,
the that's
well,
I think that's the mother of all mysteries, isn't it? I think, if you ask me.
yeah,
But
isn't
go
it?
ahead. Yeah. Yeah.
Yeah, and so I'm very interested in doing all the simulations and such, but ultimately to me it's all related to this
Yeah.
main question, so elephant in the room, which is like, well, what gives rise to the mind and the feelings and the consciousness.
Mm. Mm.
And then, of course, having spent 10 years working with and being mentored by Christoph Koch, is just
Mm.
like probably unavoidable that it rubs on you.
Yeah. Diffuses over. Yeah.
Yes,
Yeah.
which I'm very grateful for.
Yeah.
So anyway, and then it's also connects in an interesting way to what you mentioned in the beginning, or asked me about my background. So working with in physics and in quantum mechanics, where I got exposed to these ideas of entanglement, for example, right? Quantum entanglement. So, in any case, sort of bringing it all together, I published a paper last year. That's my only so far one author paper,
Okay.
published in the journal Entropy. And there I propose a hypothesis. I don't call it a theory, it's a
Mm.
hypothesis about what consciousness might be from the physics point of view.
Mm.
And so my hypothesis is that it has to do with non-separability,
Mm.
which is a concept related to entanglement, but entanglement is a purely quantum phenomenon, whereas non-separability can be a classical phenomenon as
Mm.
well.
So, what is non-separability? That's.
Yeah, essentially, it's when you have multiple degrees of freedom in your
Mm.
system, right? If some of those degrees of freedom are non-independent from each other,
Mm. So, for
that
example, it could be like the velocity in two different directions. That's two degrees of freedoms. Is that?
that could be, yes, but you can
And
talk about also multiple particles, for example,
yeah. Okay.
and in a interacting system of particles, they all affect each other, right?
Mm.
And you cannot take one particle and describe it, let's say, with a wave function that will just have the coordinates of that particle,
Okay.
right? That just won't work. You need to have a wave function that depends on multiple coordinates of all of those particles, and so such a case is non-separable. But then there are nuances, because even with interacting systems, it turns out you can often reduce them to some closely separable cases and so on. Uh, and so the hypothesis then is that the amount of this non-separability has to do with consciousness. The more non-separability you have in the system, the stronger it is, the more
Mm.
conscious the system is. And there's actually some interesting parallels between that and the integrated information theory of consciousness from
Yeah.
Giulio Tononi and others.
Cool. On that, yeah, it's fun to think about at least. So, and I think it's cool that we come out with a bold hypothesis. Also. So, thanks a lot, Anton, for coming to the podcast. It's been a pleasure to have you here.
It's my pleasure too, Gaute. Thank you very much. It was fun.